Hello, dear participant. We are in SWAM course. In course name is Introductory Animal Physiology. Module is Reproductive Physiology of Female. Title: Ovum Transport, Capacitation, Fertilization, Placenta and Parturition in Animal. Hi, I am Dr. Manish Kumar, Assistant Professor in the Department of Veterinary Physiology and Biochemistry, Faculty of Veterinary and Animal Sciences, Institute of Agricultural Sciences, Rajiv Gandhi South Campus, BHU, Barkacha. Overview of the today's topic is ovum transport and spermatozoa transport, capacitation of the spermatozoa, fertilization, implantation, maternal recognition of pregnancy, placenta, classification of placenta, and the parturition in the domestic animals. So, we will be talking of each topic one by one. First, we will start with the transport of oocytes. Transport of oocytes starts once ovulation is taking place in the ovary. So, that at ovulation, the fimbriated end of the infundibulum picks up the ovum and down picks up the ovum is transported into the uterus by combined action of the cilia and contraction of the uterine tubes. The contraction in turns are dependent on the ratio of hormone, that is estrogen and progesterone, and then hormone, that is a prostaglandin, and then signals from the autonomic nervous system. So, these three factors involves the transportation of the ova from the site of ovulation to the uterus. Immediately following the ovulation, the ovum with the vitelline membrane is surrounded by a thin zona pellucida and by a variable amount of granulosa cells, cumulus operus. A well-defined corona radiata is seen in the bitches but absent in the cow, ewes, sow and mare. The rate of transport of the ova through the ampulla portion of the oviduct to the ampullary isthmus junction, which is the site of fertilization, is quite rapid and occurs within few minutes, that is around 8 to 30 minutes in the different animals. Cumulus and the corona cells around the zona pellucida of the ovum in the most domestic animals only persist for a few hours after ovulation and fall away by ciliary and muscular activity of the oviduct. Inside the zona pellucida is a vitelline membrane. The zona pellucida is homogeneous semi-permeable structure surrounding the ovum composed of conjugated protein capable of being dissolved by the proteolytic enzymes such as trypsin and chymotrypsin. After fertilization, the vitellus and the perivitelline space is formed between the zona pellucida and the vitelline membrane into which the polar bodies are extruded. In cow, ewes and sow, the ovum is released as a secondary oocytes. The first polar body has been formed. The second maturation division occurs only after fertilization by the spermatozoa. In the case of the horse, dog and fox, oocytes ovulate as a primary oocytes, that is before the formation of the first polar body, and then mature in the uterine tube. So, this is the fertile life of gametes in the domestic animal is relatively short, and for example, if you see that sperm of the cattle, it is having the life in the reproductive tract, it is around 30 to 48 hours and ovum 20 to 24 hours in the female reproductive tract, and the different animals lifespan is given. Now, coming to the transport of the spermatozoa. Once spermatozoa is ejaculated or inseminated into the female reproductive tract, it is being transported by the contraction of the uterus towards the site of fertilization, that is ampullary isthmus junction, with the contraction taking place with the help of oxytocin at the time of coitus by the presence of the prostaglandin in the semen. Another factor that assists in the transport is thought to be the presence of the negative pressure in the uterine tube or uterus, that at that time there will be negative pressure through which also helps in the transportation of the spermatozoa into the uterus. Many spermatozoa are rapidly transported to the uterine tube after ejaculation, but it is believed that those spermatozoa are not one which is taking part in the fertilization. The spermatozoa which is taking part in the fertilization are being moved slowly later on, but the spermatozoa which is moving very fast and first, they are involved in the dissolution of the mucous membrane which is present on the tract. The spermatozoa destined for fertilization are transported more slowly from their site of deposition to the spermatozoa reservoir. The cervix of the ruminants has the prominent rhesus and mucosal crypts that provides an extensive secretory surface. The cervical crypts and their mucus covering aids in the physical barrier of the spermatozoa and serve as a spermatozoa reservoir. So that means cervix is the site where the spermatozoa are stored. Another important spermatozoa reservoir is located at the junction of the uterine horns with the uterine tubes. The spermatozoa disappear from other areas of the genital tract because of the phagocytosis by the leukocytes and macrophages. Many are trapped in the cervix. Other sperm reservoirs are located in the oviduct near the uterotubal junction. The transport of the spermatozoa from the vagina through the cervix and uterus into the oviduct is a very rapid, and it takes less than 15 minutes in all domestic animals. The cervix is considered to be the one of the ideal site for the spermatozoal reservoir. Now coming to the capacitation. Capacitation is biological significance of the capacitation was first revealed by the Austin and Chang in 1951. And capacitation means that spermatozoa have to gain the ability to fertilize the ovum. Newly or freshly ejaculated semen or inseminated semen does not have the ability to fertilize the ovum. It has to go through some physicochemical changes in the reproductive tract of the female in order to fertilize the ova. Capacitation begins in the uterus and is completed in the oviduct. The non-mammalian spermatozoa do not require this capacitation and are ready to fertilize oocytes immediately after ejaculation in the reproductive tract of the non-mammalian animals. One important changes involves the acrosome, in which the channels are established for the escape of the hyaluronidase and proteolytic enzymes. These substances are essential for penetration of the ovum. The capacitation involves the removal or modification of stabilizers or protective coats from the sperm plasma membrane. These stabilizers are also called as decapacitation factor or anti-fertility factor. That means if these factors are not removed, that in particular those spermatozoa may not be able to fertilize the ova. The capacitation is more effective during the estrous phase of the ovarian cycle than during the progesterone phase. This means it indicates the estrogen is helping in the capacitation process, whereas the progesterone is inhibiting the capacitation. In vivo, interspecies sperm capacitation is possible, which suggests that decapacitation factor are not the species specific. So that means any species of spermatozoa can be decapacitated, or capacitation can happen in the reproductive tract of the different species of the animals. Coming to the fertilization. Fertilization is the fusion of the male and female gametes to form one single cells, that is the zygote. Fertilization takes place in the ampullary isthmus junctions of the oviduct. The first step in the fertilization is the penetration of the zona pellucida by the spermatozoa. This involves not only the enzyme hyaluronidase and acrosine, but also the spermatozoon motility, means motility of the spermatozoa are also required for that. Motility ceases once contact with the oocytes has been made. In most domestic species, the second maturation division that occurs when the spermatozoa penetrates the zona pellucida, whereas the first meiosis occurred a few hours before ovulation. The zona reaction occurs after penetration of the zona pellucida and protects the oocytes from further penetration by the other spermatozoa. Penetration by the more than one spermatozoon is called as polyspermy is deleterious to the normal development of the zygote. Pronuclei develop from the nuclei of the spermatozoon and oocytes, which is followed by fusion of respective pronuclei to form the zygote with the diploid number of chromosome. Fertilization is complete after the fused pronuclei have disappeared and are replaced by by chromosome groups united in prophase of the first mitotic division. Zygote usually remains in the uterine tubes for 3 to 4 days before being transferred to the uterus. Progesterone has the quickening influence on the uterus and promotes the development of the glandular endometrium that can secrete the uterine milk, which is very important for the nourishment of the embryos. The cell division proceeds a 16 to 32 cells structure known as morula, a cavity formed within the morula by 6 to 8 days of the age, and the cell mass is called as blastocyst. The period of oocytes ends when the blastocyst attached to the endometrium. This is the beginning of the embryonic period. So once the blastocyst attached to the endometrium, so this is the time where the this embryonic periods of is starting. The embryonic period is characterized by the rapid growth, major tissue, organs, system develop, and major features of external body forms become recognizable. The fetal period extends from this time until birth and begins about 45 days of the gestation in the cow. Now the block to the polyospermy. That means that it is the process by which once first spermatozoa penetrate the oocytes, it takes some chemical reactions are taking place at the zona pellucida, and it doesn't allow the second spermatozoa to enter, which is deleterious for the embryonic development. Immediately following the fertilization, the ovum surface changes and prevents further fusion of the additional spermatozoa. The block to polyospermy, that is entry of the multiple spermatozoa, is at the zona pellucida in sheep and swine, with the secondary physiological block at the vitelline membrane in the case of some species, for example, it is a rabbit. Initiation of the block occurs at the point of the sperm penetration into the ovum, where the central granules are released into the perivitelline space, which causes the excessive reorganization of the zona pellucida and vitelline surface, and this is referred to as the zona reaction or cortical reaction. This causes the release of the enzyme to harden the zona pellucida and thus inactivates the sperm receptor, that is a ZP3 receptor, which further prevents the entry of the supplementary or extra spermatozoa to the zona. Now coming to the implantation. Implantation is the process whereby the new embryos become established at the developmental site on the endometrium to develop into the fetus. The nutritive requirement of the developing blastocysts are satisfied by the diffusion from the yolk in the oocytes and by secretion of the uterine tubes and uterus, which is called as uterine milk, until it becomes fixed in a position in the uterus. Implantation of the embryo occurs when it becomes fixed in a position and forms a physical and functional contact with the uterus. It occurs 2 to 5 weeks after fertilization in almost all the domestic animals. Following fertilization, the new embryo reaches the uterus 4 days post-estrus in the cows and ewes. The nutritive requirement of the embryo are supplied by the yolk material and secretions of the oviduct ducts and uterus. The embryo remains free-floating in the uterine fluid after its entry into the uterus until implantation and derives its nutrition from the endometrial secretion. Because the embryo continues to grow, the central mass of the cell becomes further removed from the surface. The embryo continues to grow by mitotic division and the cells become arranged to one side, that is known as compaction, and the fluid accumulates in a central cavity known as blastocele, and the conceptus is called as blastocyst. The blastocyst has two cell type, that is, one is trophoblast, which later on forms the placenta, and the inner cell mass which is develops into the fetus. When the blastocyst reaches the uterus, it is still covered by the zona pellucida. In 6 to 11 days after fertilization, the zona pellucida breaks down and releases the embryo, which is referred to as zona hatching. Zona hatching is contributed by the blastocyst expansion and contraction added by the lytic enzymes. Zona hatching in the cattle, it takes around 9 to 11 days, in horse around 8 days, sheep 7 to 8 days, swine 6 days and days after fertilization. The blastocyst elongates because of the formation and hyperplasia of the extraembryonic membranes. Blastocyst elongation occurs in cows, sheep and pigs, but not in the horse. The concurrent with the blastocyst elongation, gastrulation occurs in the embryo, that is formation of the ectoderm and mesoderm and endoderm. A spacing of the embryo in the uterine horn in the Polyptychus animals occurs during this period. In the litter-bearing animals, transuterine migration of the zygote helps in the distribution of the embryo, which is affected by the uterine muscles contraction in the presence of the embryos. So, this spacing of the embryo in the different Polyptychus animal is very important in order for their proper growth and development of the fetus in the uterus. So, coming to the types of implantation, there are two types of implantation. One is invasive and second one is non-invasive. Invasive types of implantation is taking place in the primates and rodents, where the blastocyst penetrates the uterine epithelium. This is accomplished by the proliferation of the uterine epithelium in the vicinity of the blastocyst, and non-invasive implantation normally takes place in the ruminants, horse, pig, etc. Non-penetrating and it is a superficial. It involves the absorption, opposition and addition of the blastocyst and uterine epithelium, or integration of trophoblastic villi and uterine epithelium. Now, coming to the maternal recognition of pregnancy. Maternal recognition of the pregnancy is very important in the case of domestic animal, because this is the time where that signals from the uterus are being sent to for the continuation of the CL, or that that conceptus will develop, and PGF2 alpha from the placenta will not be secreted, and lysis of the corpus luteum will not take place. It will continue the production of the progesterone, endometrial development and secretory activity will be continued. This critical period, this is a critical period when the conceptus gives the signals, is called as maternal recognition of the pregnancy. In cow, around 16 to 19 days of the pregnancy, conceptus secretes the bovine trophoblast protein 1, and conceptus move 12 to 14 times per day between 12 to 14 days of the gastrulation to limit the PGF2 alpha production from the uterus. In sheep, secretory protein, that is ovine trophoblast protein or type 1 conceptus interferons are one which is responsible for the sending the signals for the recognition of pregnancy. So, now coming to the period of pregnancy, that prenatal life of the individual is divided into 3 periods, that is a period of ovum, period of embryo and period of fetus. Period of ovum means it is period between the fertilization to the blastocyst formation, and period of embryo is blastocyst development to the organogenesis, and the fetus is means its time during which the most part of the placenta fetus develops and lasts until the parturition. This is the period of pregnancy in the different domestic animals. In the cattle, if you take, it is around 280 days, and buffalo it is 310 days, and so on. In the different animal, like cat, it is 50 to 65 days is the gestation period. Now coming to the placenta. As the embryo increases in size, the extra embryonic or fetal membrane of the placenta develops to meet the increasing need for the more nutrients to nourish the developing embryo. Because once fetus will start developing, that embryo, as it is taking the nutrients, all the nutrients from the uterine milks, so as the development takes place, that uterine milk will not be sufficient to nourish the developing embryo. So that placentation need to take place, and formation of the placenta taking place, through which all the nutrients are being supplied to the fetus. The trophoblastic cells of the embryo participate in the formation of the placenta. The outer single layer of the cells of the blastocyst, refers to as the trophoectoderm, contributes to the outer layer of the placenta. Inside the trophoectoderm is a group of cells called the inner cell mass, which forms the embryo proper, also a portion of the placenta. The fetal membrane consists of the primitive yolk sac, amnion, allantois, and chorion. The chorion is the outermost membrane and is in contact with the maternal uterine endometrium. Chorioallantoisis or allantochorion is formed by the fusion of the outer layer of the vascular allantois to chorion. The chorioallantois or allantochorion is formed by the fusion of the outer layer of vascular allantois and chorion. This structure, richly supplied with the blood vessels communicating with the endometrium, carries the metabolic interchanges of the gases, nutrients, waste between the fetal and maternal circulation. And the allantois arise in the second and third week of gestation in the bovine fetus. The allantois cavity stores the waste product of the fetal kidneys. The allantoic sac, a fluid-filled space formed by two layers of the allantois, is located between the amnion and the chorion, is called as first water bag. Amniotic sac. Amnion forms about 13 to 16 days after the conception in the sheep and cows. It is a double-walled sac, completely surround the fetus except at the umbilical ring. The amniotic cavity play a role in the care of some waste product. The yolk sac, it is a primitive structure developing early in the embryonic period and disappearing after a short period of time in ruminants and swine, but persist 4 to 6 weeks in the horse. Now coming to the classification of the placenta. On the basis of the layer present in the placenta, it has been classified into the different group. Maternal tissue is having three tissue, that is endothelium, connective tissue, and epithelium. Fetal side also have chorion, connective tissue, and endothelium. All these 6 layers together barrier to separate the maternal blood from the fetal blood. So based on the number of layer, the placenta has been divided into the epitheliochorial. It is found in the mare, sow, cow, and donkey. Syndesmochorial, it is found in the eve and does. Endotheliochorial, it is found in the bitch, where the endothelium is the only layer separates from the maternal sides. And hemochorial, it is found in the monkey and omen, and it is the all the 3 layers are disappeared from the maternal side. Hemoendothelial cells, it is found in the guinea pig, rabbit, and rats. It is having the mother side, there is no layer, and from the fetal side only endothelium will be present. So on the basis of the loss of the maternal tissue at the time of parturition, placenta are classified into the 2 types, that is a deciduate or non-deciduate. Deciduate type of placenta is found in the bitch and queen, monkey and omen, means during that at the time of parturition or the time of birth, maternal tissue are being lost. Non-deciduate types, in this type of placenta, at the time of parturition, that maternal tissue doesn't lost at the time of birth. It is found in the sow, mare, ewes, doe, and cow. So on the basis of the chorionic villi pattern and the shape of attachment to the uterus, the placenta are classified into the diffuse type of placenta, means this is scattered all over the surface of the chorion. That villi will be scattered all over the surface of the chorion, and this type of placenta are formed in the horse, pig, and camel. Quaternary type or multiplex placenta, it is grouped into the means villi will be grouped into the multiple circumscribed area, and it is found in the cow, sheep and goat. Zonary type, it is found in the goat and cat, means it is quaternary villi will be attached in the zone like area. And discoid means it is arranged on the large flattened area in the form of a circular plate or disc like things. It is found in the primate and rodents, and bidiscoid it is found in the monkey. So this is again the elaboration of this type of placenta. So if coming to the function of the placenta, placenta aids in the transport of the nutrients such as carbohydrate, protein, fatty acid, glycerol, water, inorganic salt, but lower concentration of the vitamin A, D and E from the mother to the fetus. Fructose comprises of the 70 to 80% of the sugar in the fetal blood, and it is produced by the placenta from the glucose. It is involved in the gaseous exchange between the mother and the fetus. It is also act as an excretory organ for the fetus. It is a temporary endocrine organ. It secretes the equine chorionic gonadotropin and human chorionic gonadotropin, and it helps in the parturition at the time of parturition. It helps in the maintenance of the pregnancy and during the pregnancy, and at the time of parturition it helps in the contraction and expulsion of the fetus. So now let's talk about the parturition. Parturition is nothing but it is a delivery of the fetus and fetal membrane from the mother at the time of once gestation is completed. The parturition is divided into three different stages, that is the dilatation of the cervical canal to accommodate the passage of the fetus from the uterus through the vagina and to the outside. Contraction of the uterine myometrium that are sufficiently strong to expel the fetus and the expulsion of the placenta. So this is the three different stages of the parturition. If you see the sign of the parturition, what are the different signs of the parturition will be there? Several events takes place near the end of the pregnancy in the preparation of the parturition. As parturition approaches, the cervix begins to soften. It is the ripening of the cervix, it is called as a result of the dissociation of its tough connective tissue, collagen fibers, which is packed during the pregnancy. Most sign of approaching parturition is related to change in the pelvic ligament, enlargement, edema of the vulva and mammary activity. Physical signs: throughout the pregnancy, the abdomen continues to enlarge and its maximum size reaches just before the parturition. Mammary gland also continues to enlarge and within a few days of the parturition begins to secrete the material that is called as waxing. The waxing is most common in the case of the horse, and it is observed 6 to 48 hours before the foaling, and it is replaced by the drip of milk or a stream of the milk 12 to 24 hours later. There is a swelling in the vulva, discharge of the mucus, animal will abdominal muscles will relax, belly will drop down, ramp will sink on the both side, respiratory rate will increase steadily in the sow. Hormone relaxin is associated with the increasing level of the estrogen in the late pregnancy, causes the relaxation of ligaments and to enable the birth canal to enlarge. PGF12 helps to relax the cervix. Rectal temperature decreases also indicate the impending parturition. Progesterone increases the basal body temperature because causes the increase in the basal metabolic rate, whereas however, once the corpus luteum will regress and the progesterone concentration will fall down, estrogen concentration will increase, so that body temperature will decrease, and this is the decrease in the body temperature is a most reliable sign of the parturition, approaching the parturition in the case of bitch, and the body temperature falls around 2 to 3 degrees Celsius and 4 to 5 degrees Fahrenheit in the case of the bitch. In the other domestic animal, fall in the temperature is not the reliable sign of the parturition in the other domestic animals. So if you talk about the behavioral signs of the parturition in the domestic animal, the frequent urination will be there, animal will be restless, animal will be getting up and lying down quickly, frequently, and polytocous animal which is giving birth to more than one young kids or young one, they will have the nesting behavior for the parturition, before the parturition. So mechanism of parturition, if you talk about, the successful parturition depends on the myometrial contraction and the dilatation of the cervix. So myometrial contraction means the contraction of the uterus during the gestation is blocked because of the progesterone. As the parturition approaches, the progesterone concentration decreases and estrogen increases, PGF-12 increases, that causes the contraction and help in the parturition. Dilatation of the cervix is very important because that birth canal need to provide the space for the passage of the fetus through the cervix. Initiation of the parturition is triggered by the fetus and completed by the complex interaction of the endocrine, neural and mechanical factor. Hormonal changes taking place is important. Hormones like production of estrogen is an important one for the helping in the parturition. Progesterone concentration decreases. Fetoplacental unit produces the estrogen 3 to 4 weeks before parturition in the cows. An increase in the production of the cortisol by the fetal adrenal cortex that concurrent with the maturity of the fetus and initiate the prepartum increase in the estrogen production. The secretion of the estrogen assists in the production of the uterine muscles contractile protein. Before parturition, estrogen might also be signals for the secretion of the PGF2 alpha. PGF2 alpha initiate the regression of the CL if it is present, and the increase in the estrogen and decrease in the progesterone levels convert the uterus from the state of quiescence to the state of contractility. However, PGF2 alpha level increases during the foaling. The progesterone concentration does not decrease in the mare PGF2 alpha secretion because no CL present, and after around 150 days of the pregnancy show that increase in the contractility of the uterus permitting the greater mobility of the sarcoplasmic reticulum by the PGF2 alpha. These early contraction increases might be important in positioning the fetus for the delivery and presentation through the pelvic canal. So, the presence of the fetus in the cervical canal causes the oxytocin to release, which is again helping in the parturition, and the presence of the feet in the pelvic canal of the fetus is a consequent stimulus of the vagina provided by the reflex contraction of the abdominal cavity. This is also called as the Ferguson's reflex. Ripening of the cervix is that again that it is being done by the estrogen, PGF2 alpha, relaxin, and initiated by the degradation of the corpus luteum. But placental dependent species like horse, fetal cortisol activates the 17 alpha hydroxylase. It converts the progesterone to the estrogen. The rising concentration of the estrogen and declining concentration of the progesterone stimulates the PGF2 alpha secretion. PGF2 alpha plays a central role in inducing the strong uterine contraction of the second stage of the labor and causing the dilatation. Ferguson's reflex is taking place when the feet of the fetus is coming into the birth canal, and which helps in the secretion and contraction of the uterine muscles and helps in the parturition of the fetus. So, that greatest effort is associated with the emergence of the head and chest into the birth canal. The expulsion of the placenta is rapid in the mare but slower in the ruminant due to the cotyledonary type of the placenta. In the pigs, contraction begins at both the ends of the uterine horns and subsequently are propagated forward the cervix or opposite direction. The largest mass of the placenta, however, is usually expelled 3 to 4 hours after the delivery of the last piglet in the case of swine. If you see this flow chart, it is very clear that how the fetal cortisol and the estrogen production, PGF2 alpha, regression of the CL, decrease of the progesterone secretion, and all together leads through myometrial contraction and the expulsion of the fetus is taking place. A stage of parturition, it is divided into 3 stages. In the first stage, the contraction of the uterine muscles will be there, happening of the cervix will be there, dilatation of the cervix will be there, so that discharge of the mucous membrane will be there, birth canal will be softened and it will be lubricated by the mucus secretions, and it will last around 2 to 5 hours and up to 12 hours in the beach. And stage second, it is the entrance of the fetus into the dilated birth canal, and Ferguson's reflex will take place, and contraction of the uterus and abdominal muscles will be there, and expulsion of the fetus will be taking place in the stage second. And in stage third, it includes the expulsion of the fetal membrane or placenta. So this is the stage of stage of the parturition. And this, you can see the table where the stage of labor and related events in the farm animals is given, and how much time it is taking, and what are the things, what are the events it is taking place for the parturition of the animal. With this, we are covering the complete reproduction of the female animals. Thank you.